So it's finally happened. Sort of. This video is going to be on my YouTube channel, Zenthanol, like all of my Zenthanol live videos are. But I wanted to talk about hashtags today. Much love, Major CBD, Sean Major. What's up? I'm going to be talking about the use of hashtags and how I utilize hashtags for science communication reasons. But the thing that I'm referring to that has finally happened, kind of, is that I finally had a post that I felt was a really good representation of the philosophy that I use when it comes to utilizing hashtags. When hashtags were first introduced as a thing, as a concept, I did not like them. I basically found them sort of like a joke, right? Like a lot of people do, people using them way too much, or people using them perhaps in a way that's kind of, I don't know, exploitative or kind of fake or something. I don't know. But what I do know is that the way that I utilize hashtags is kind of at least on Instagram and somewhat on other platforms, it allows somebody to search for the scientific or the binomial names that I often hashtag, or the families, taxonomy in particular. People are able to click on Arabidopsis, or they're able to Arabidopsis, I should say, or one of the many different technical taxonomies that I put in my posts, and they might be like, I wonder what Hymenoptera is, or I wonder what is Coleoptera. Well, those are beetles, and the other one is bees, wasps, and ants, or Hymenoptera. So, basically, the idea is to use it kind of like a dictionary, a little bit. In the video that I'm going to put up when I edit this out and put it on YouTube, I'm going to have pictures that kind of show what I'm talking about. But I have here 4 sort of examples. I have a primary post, which is the one that I posted right now. Not right now. What I posted today, actually, which was about the research report called Characterizing Both Bacteria and Fungi Improves Understanding of Arabidopsis Root Microbiome, or something like that. Anyways, so in it, we talked about a whole bunch of different kind of taxonomic groups from fungi and bacteria, obviously. And as I was going through the hashtags, I thought to myself, I wonder if I can find some of these posts if I just click on the hashtag, because some of these things probably people aren't talking about, because they're really sort of esoteric. And I've talked enough about microbes and fungi and bacteria with regards to pests and pathogens and plant physiology and that kind of stuff in general, that it sort of worked out. And that's what I mean by a good example. I've had plenty of other posts that are kind of like that. And about a month ago, I was shadow banned, actually, which came to a surprise, but came as a surprise. However, I'm not super surprised, because some of the nature of what I talk about has to do with cannabis, and that can be sort of contentious in some social media settings, right? And I have to admit, I was a little bit hurt, but it went away in 3 weeks and everything was fine. But one of the things I didn't like was because I like to be able to click on a hashtag and be able to look at my other posts in that group, So that's all the context I have. Let me sort of explain what I'm talking about. So essentially, when I do hashtags, I usually hashtag one of 3 things. I'll hashtag a taxonomic group, so a species name, and I won't put it together. I'll separate it usually. And the reason why I do that, there's 2 sorts of camps, I suppose, or at least for me, there's 2 rationales. On the one side, you could put the whole name together as one thing and make it much more unique, and more likely that people are only going to be coming across the species that you're talking about. And that makes a lot of sense to me. But I don't do that. What I do is I have the genus name and I have the species name separated. So I'll hashtag or link or whatever the genus and then the species separately. I will also do other taxonomic ranks. I'll also do important keywords as well. And the reason for this is because the keywords might be really important to kind of bring a whole bunch of different posts together under one umbrella. So, like, for example, and they have to be pretty unique. You can't pick something that a lot of other people are hashtagging because then you won't come across those educational posts, the ones that I'm posting. And I guess a lot of this is sort of specific to me. I don't know anyone else who's like doing this in the same way that I am, but I mean, there's a lot of people on social media. I'm not surprised when people do. When I do click on a species name that's very common, and it also might be common with other kinds of animals. So I might be referring to some insect with the species name of I don't know, like bipuncta, which means like two punctures or like two dimples. But there might be tons of other organisms with the species name bipuncta, but a different genus, right? So you click on bipuncta and you'll get all of the bipunctas. Or if you click on, I don't know, some other thing, crotalus for like rattlesnakes or whatever, then you'll get rattlesnakes of all kinds, right? And then if people are just posting crotalus in general for whatever reason, maybe they're like a herpetology post or an account or something, they might put crotalus on things that aren't crotalus. So you just got to be careful with that kind of a thing, I suppose. But with keywords, I think it's really helpful. Like melanization, for example. So that's a process that happens in plants, it's a process that happens in insects, and it helps them to mitigate certain problems. So why is that helpful? Well, because if I want to talk about melanization in general, then I can just click on melanization hashtag and find all of my posts and other people's posts where they've talked about that particular process. Or, in some cases, it gets co-opted by other organizations or something. For example, if I use what's the word? Like an acronym or initialism. So like USDA. Usually the posts have to do with the United States Department of Agriculture, but not always. I know that the CDFA, the California Department of Food and Agriculture, that's usually going to be what I find, but not always. And like Europeans might have a different CDFA, so that might get more popular. And if people don't post about it, then it doesn't come up as much, right? So it's not a foolproof system by any means, but it's helpful, and I like it. So here are my examples. You won't be able to see them, but when I edit the video, you will be able to see them. So check it out if you want to on my YouTube channel, Zenthanol. So the post I made today, I'm just going to read it verbatim. I type, this is mostly from the abstract. Despite their close physical proximity, researchers in characterizing both bacteria and fungi improves understanding of the Arabidopsis root microbiome. So I hashtag Arabidopsis, which is a genus name, and microbiome, because that's a term that I think is really important. So I can associate this post with the microbiome, a big part of the paper. That makes sense, right? So they found that the leaves and the roots of Arabidopsis thaliana. So then I find the species name and I hashtag the species name, right? So now we have Arabidopsis, the genus, and Thaliana, the species. Although they weren't right next to each other, they are both linked. So somebody can click on the genus for Arabidopsis or Thaliana for the thale cress, Arabidopsis thaliana. They found that it differed in the composition of the microbial communities. As an example, roots contain a lower proportion of, and here I hashtag the word, Proteobacteria, because that's a group name, than leaves, and a correspondingly higher proportion of the phyla, and here I hashtag Actinobacteria. Bacteroides, Bacteroidetes, and Chloroflexi. So all three of those complex names I hashtagged because, one, not a lot of people hashtag those names, so the posts that I make are going to show up about that. Plus, I have a private account, so it's not public to other people, so I can at least reference back to posts that I've made and other people have made publicly, although since most people don't publicly make posts about it, it's kind of like my own personal digital reference book, if that makes sense. In the case of fungi, Ascomycota and Basidiomycota are more common in the leaf than root microbiome, whereas members of and here I hashtagged Ascomycota and Basidiomycota, but here I also hashtagged, I don't even know this one, usually I'm not stumped, Mordirellomycota, possibly, are moderately enriched in the root microbiome. Differences among leaves and roots are more pronounced at increasingly specific taxonomic levels. For example, bacterial genera enriched in the root microbiome include, and here comes a whole bunch, all the complicated names are hashtagged here, Massilia, Flavobacterium, and Actinoplanes, whereas Pseudomonas, Xanthinobacterium, and Sphingomonas are more common in the leaf. Interesting to note, when I went to click on Actinoplanes, Instagram let me know that apparently it's been banned, I guess. I guess people associate it, or at least Instagram associates it with posts that are, not in keeping with their terms of service or something. I don't know how that's possible, unless, there's a bunch of, gore, perhaps. I've seen some really terrible fusarial outcomes from, humans and animals that are infected by fusarial diseases. It does happen, although usually only with immunocompromised people. My point is that that's a possibility. Maybe all the posts that had to do with a certain group are mostly just to do with maybe septic conditions or really terrible things like that, which I don't want to be looking at either. But it's important medically, I suppose. Where did I leave off? Oh, in the case of fungi, Tetracladium, Mordiarella, and Paraphoma were preferentially associated with roots, whereas Alternaria, Articulospora, and Cladosporium, and Plectosphaerelia were more common in the leaf. Overall, fungi tend to be more difficult to classify at the genus level from bacteria, while microbes from both kingdoms were more difficult to classify in the root than in the leaf microbiome. On average, leaf and root bacterial communities tend to be more similar than leaf and root fungal communities. Interesting point. Research in barley has reported that the SEC61 beta gene associated with the Thale crust microbiome fungi also plays a role in leaf microbial interactions, as silencing SEC61 beta leads to an increase in plant resistance by disrupting contact with fungal haustoria. And this is a really important thing to note. And I wanted to really touch upon this and iterate this in the post. Something I've reiterated a bunch of times is that resistance in plants does not just, does not just have to do with or not resistance rather, although it does play a role. The microbiome, beneficial, the same mechanisms that allow pathogens, at least some of them, to get into a plant are the same physiological mechanisms and then lack thereof that allow beneficials into the plant. Some people know that mustard plants tend to not have a mycorrhizal interaction at all, really. They don't build those relationships. And that's possibly because, genetically speaking, they just don't have that proclivity. For whatever reason, evolutionarily. I actually don't know, personally, and I don't even know the extent of it, so it's possible that it's just that there are some that don't exist, rather than totally not possible, no documented examples. So they found here that if they silenced this gene, then the plant was more resistant to certain pathogens, and also more resistant to establishing these relationships, and that's the reason why. In fact, in some groups of microbes, we will find that there are several species of pathogens, and right along with them, there are several species of beneficials. And they both evolved from a common ancestor. Some of them became plant parasites, and some of them became beneficial to the plants. Maybe different plants, or the same kinds of plants. There's no like good or bad guys, like I want to remind people. There's no like good guy side and bad guy side. If you're a fungus, you'll be killed by fungicides. If you're a good fungus, then the cultivator wants to keep you. If you're a pathogen, then they don't necessarily want to keep you at all. The root microbiome is not only shaped by immune-related genes, but also by genes that determine plant form and physiology. Experiments to investigate the most promising candidates are currently underway. So that was a lot to say in a short amount of time. But that's kind of my point here, is that this post had a lot of different taxonomic tags. And now that you have all of that context, I can go on to the next part of this. So, I was looking through these hashtags and I said, I remember talking about this one, I remember talking about that one, I remember talking about this one. But in my experience, people don't tend to talk about these groups at all, or like hashtag them at all, which is fine. So I was curious if I would be able to find my posts really easily, and I did, several of them. And so the first one that I found was for, which one was this? For actinobacteria. Yes, okay. So this post, which I made in April 18th, 2018, I say the hashtag Rhizosphere, and more specifically the hashtag Rhizoplane, is an area of intense dynamism and complexity, where plant-microbe interaction is most prominent and diverse for most land plants. In dynamic root exudate chemistry and microbial substrate preferences drive patterns in Rhizosphere microbial community assembly, say that 5 times fast, researchers studied the interactions between wild oat Avena barbata roots, their phytochemically active rhizosphere, and soil microbes in order to better understand what is termed the Rhizosphere Effect. Simply put, this is a phenomenon wherein plants selectively encourage and discourage the microbes in their microbiome by the compounds they exude from their roots, which changes over time, sometimes dramatically, based on plant age, environment, microbial community, and other factors. Populations encouraged tend to be phylogenetically related. That means that they are from the same kind of like ancestral lineage, having similar preferences for certain exudate constituents, which sustain them and sometimes repel others. Because of this shared preferences can be predicted using genomic sequencing, and here I hashtag a bunch of words. Nicotinic, shikimic, salicylic, cinnamic, and indole-3 acetic acids encourage bacterial growth, whilst proteobacteria, specifically alpha proteobacteria, for example, Burkholderia, Pseudomonas, which are both hashtagged, and if you remember, Pseudomonas was hashtagged in the first post, seem to be commonly encouraged across many studied soil and plant systems like Zea oryza, which is rice, Zea oryza, Arabidopsis, which is also hashtagged in the other post, Triticum, actinobacteria, for example, Bacillus, Paenibacillus, seem to decrease in relation to plant age. The general trend shows a concerted link between plants and specific microbial communities. So there's a lot to like digest from that, and I kind of spoke quickly and I didn't really explain it all. But my point is that I was able to go from this post that's kind of concerning the microbiome and microbes and plants and that kind of a thing, And I was able to find another post that I made months and months in the past that kind of talks about some of the same sorts of things. And so if I wanted to make a video about that, or about those concepts, it wouldn't be hard for me to scour through my posts by simply jumping and hopping from one hashtag to the other hashtag to the other hashtag, kind of like hyperlinks on Wikipedia. I'm sure many of you who know me probably have also done the same sort of a thing, where you find an interesting topic, look it up online, find a hyperlink that tells you more information, and then you go and you lateral to like one thing and then another thing, and then you find yourself in a Wikipedia hole, and then it's 2 a.m. and you're researching something that's totally adjacent to what you were talking about. So you can also do something that's a little bit more on point and a little bit more accurate to what you're trying to talk about in this way. So that was the first thing, and I have a picture here that goes over all of these different microbes that were found to have either a negative or a positive response to root growth, which again is related to the original post that I was talking about microbes and the leaf and root microbiome. The second post, I believe, has to do with bacteroides. The second node is what I'm calling them. Was from January 5th, 2018. And the post reads, "An apt name for a pair of gene clusters, Jekyll and Hyde plant-associated genes, were one of the more interesting discoveries in genomic features of bacterial adaptation in plants. Researchers sequenced 484 genomes of bacterial isolates from roots of Brassicaceae, Populus species, and Zea mays. Comparison with 3,837 recorded bacterial genomes identified many so-called plant-associated gene clusters. Plant-associated, for example, alpha proteobacteria, bacteroidetes, Xanthomonaceae, and Pseudomonas bacteria, all of which were hashtagged, had genomes encoding more carbohydrate metabolism functions and less for mobile elements than related but non-plant associated genomes. They experimentally validated candidates from two sets of plant-associated genes, one involved in plant colonization, Jekyll, and the other serving a microbe-to-microbe competition between plant-associated bacteria, Hyde. Pathogenic and non-pathogenic bacteria of the genus Acidovorax, order Burkholderies, were found to have gene families that matched perfectly to isolate lifestyle. Non-pathogens had the Jekyll gene, and pathogens the Hyde gene. Even nearly identical non-pathogenic isolates had hypervariable Jekyll loci, the plural locus, that were substantially more divergent than neighboring genes. Hyde-carrying organisms included other phytopathogens, such as, and here I hashtag, Pseudomonas syringae. The gene often precedes type 6 secretion systems, or hashtag T6SS, again, an acronym, which are typically used in competitive interactions. Convergent evolution and horizontal transfer of protein domains from eukaryotes to bacteria have been suggested for some microbial effector proteins that are secreted into eukaryotic host cells to suppress defense and facilitate microbial proliferation. Amongst bacteroidetes, and Xanthomonaceae were these most abundant. In a related fashion, Jacqueline, or Jacqueline, is a lectin that binds to mannose, which is also hashtagged, itself found on the walls of some fungal and bacterial pathogens. Mannose may act as a microbial-associated molecular pattern, or a MAMP, which I've hashtagged. Typically, MAMPs are detected by the immune system of a plant by binding to them. Lectin secretions may bind to mannose such that the immune system cannot detect them any longer, functioning like camouflage. Microbes are varied, and that was the post. So, what am I trying to say here? So, okay, in the first in the first post I talked about, the original one that links to all these other ones, I was talking about the root and leaf microbiome. And in this post here, I also am talking about organisms that are related to the root and leaf microbiome. Obviously, because the other post mentioned them by name, or at least mentioned their group by name. And so what I'm getting at is that somebody can then, like myself, go through all of these hashtags from these taxonomic ranks and then kind of look at previous research reports, almost like, again, like a personal diary or something like this. And by doing so, if I want to talk about this sort of a thing, I might be able to find other interesting facts to add on, and I might be able to find what's most important for somebody who's in the cultivation space, patterns, and perhaps even ways to be clever with utilizing such organisms, especially when we're finding out how important the microbiome is. Epiphytes, endophytes, things that live on the top of the leaf, things that live inside the plant systemically or only translaminarly, or things that live in the root microbiome, or things that live on the rhizoplane or the rhizosphere and all that sort of stuff. Third node. This one's a little bit... Nope, actually it's just as long as the other ones. This post, it was from April 25th, 2018. And this one is kind of cool because whereas the other ones talked about what could be potentially beneficial and detrimental organisms from the plants, or from the microbes for the plants rather, this one talks about microbes that could be detrimental to pollinators, specifically honeybees. The honeybee Apis mellifera, both the genus and the species is hashtagged in this post, is perhaps the most popular livestock species in the world as far as media attraction goes. Many less people talking about saving the cows, which is a funny quip by me, which is essentially talking about how honeybees are about as likely to go extinct as cattle. They're basically livestock. They are livestock, honestly. They're micro-livestock. So the idea of honeybees, which are, by the way, not from North America, there are no honeybees that are native to North or South America. They're from Southeast Asia, though they have expanded with human culture and climate change and things like that. But, I mean, that's kind of my point, is that they're not from here. So it's not surprising that they would have some potentially detrimental effects on other ecologies, as most invasives do. As I say in the post, it is understood that honeybees can pick up microbes through pollination practices and nectar feeding, including those associated with other pollinators like bombus species. I've hashtagged bombus as the genus. Bombus are bumblebees. While their gut microbiome, which I've hashtagged, is also a subject of research I made a typo there. In order to better care for them, researchers took a look at hive microbiota in bacterial communities associated with honeybee food stores are correlated with land use. As is the case with most arthropod microbe interactions, spatial layout and topography was an influence. Stored pollen, often referred to as bee bread, was found to include the genera Pseudomonas, 32.4%, Arsenophonus, 13%, and Acinetobacter. Yeah, Acinetobacter, 5.2%. In average, which in total accounted for 66.48% of the sequences generated within this study. Cultivators will notice several of these genera are associated with phytopathogens. Other genera were present in all 48 bee-bred samples, including the already mentioned Pseudomonas, Arsenophonus, Lactobacillus, Erwinia, and Acinetobacter, as well as the, and I hashtag these, Saccharobacter, Rauwtella, Tatumella, Massilia, which you might remember from everything else I've said so far with the other posts, Orbis, and Sphingomonas, accounting for less than 2% of sequences in total. Knowing this information may help apiarists and agricultural entomologists in the constant battle to keep honeybees and other anthophila bees happy. So, in this third node, in this third picture, in this third post that I've made, I am making a very direct correlation, and I'm drawing the connection between these microbes that are found on plants just in general, and their association with honeybees in this case, but also bumblebees and other organisms that feed on pollen and nectar on plants. So why is this important? Well, it's really important if you're trying to figure out how different phytopathogens might pass from plant to plant. Certainly, it's not outside of the realm of population. Certainly not outside the realm of possibility and plausibility that phytopathogens can be carried on a pollinator that would be going from one plant to another plant. So a phytopathogen, especially one that's passed through pollen, for example, which there aren't a whole lot of, but they do exist, like a virus, for example, could be in the pollen of the plant and then get carried from a honeybee to another plant, even a plant that doesn't necessarily receive the pollen that has been contaminated. It might be a different species or be incompatible in some way. But if it wasn't, then that virus is going to get into that plant, and now you have another plant that is infected by the virus. And possibly even the seed stock, too, being vertically transmitted. So that's just a thing to consider. And I'm not saying that it's like this next big problem. I'm not trying to make that sort of a statement. But I do think that it is an interesting concept to consider because the more we learn about how complex the microbiome is with plants and farming and trying to encourage those relationships between beneficial bacteria, beneficial fungi, beneficial microbes in general, microarthropods, biocontrol agents, all of those things. Trying to encourage those relationships is tricky and complex and depends on the context of your cultivation, what plants you're growing, a bunch of other factors, where you live, all of that. And it's never going to be less complex than that. Even if you grow in a monoculture and that sort of a thing, you will always be susceptible, at the very least, to, I guess you could say, changes in ecology, right? Whether you planned for them or not, sometimes they just happen. And knowing what's possible is a great way to defend yourself against the mysterious and the things that have yet to happen. Alright, last node, last post. This was from January 23rd, 2018. And this talks about one of my favorite subjects, the holobiont and the hollow genome. It reads, the holobiont is the combined aggregate of all the species in a particular ecological unit. Individual plants and animals are themselves ecological units, and every individual associated with this unit is a biont, with the entire genetic aggregate being the hollow genome, which I have hashtagged. I also hashtagged holobiont. Why? Because they are keywords that are really important to the point of the post, and also they tend to be things that people don't really talk about. So when I use the hashtag holobiont, I'm probably going to find my own posts and not other people's posts, simply because other people aren't hashtagging in the same way. I kind of wish that they did, because then I would have other interesting things to research and look up, and other people who are using that hashtag, which is how hashtags work. It's definitely not, I'm not inventing how hashtags work, obviously, but I think that they are not necessarily considered, I don't know, people don't think about them in science communication, I think, in the way that they could. Plant seeds can, and often do, contain beneficial or detrimental plant genotype-specific microbes. I'm going to pause here. So that's another important point that was brought up in previous posts that I made in this series. Additionally, and most specifically in the first one, because we're learning that the microbial interaction with host plants, whether they're pathogens or non-pathogens, whether they're mutualistic or commensalic or parasitic, however they are aligned, however we categorize them as good or bad or neutral. A lot of times those interactions are mediated by a very, very specific gene-to-gene relationship or multiple gene-to-multiple gene relationship. So much so, and a lot of it has to do with the immune system of the plant. So much so that if the plant is particularly robust and if it has certain pathways of immunity, beneficial microbes won't even be able to make the sort of interactions that they're used to. And in case for those who aren't already aware, people didn't know what microbes were back in ye old days when they were breeding for plants to try to make the fruits bigger or make the plants more hardier, grow faster, or take the tallest ones or whatever we're doing here. They didn't know about that stuff. So, unsurprisingly, wild versions or wild populations of cultivated plants, cultivars that we grow nowadays that have been standardized, they tend to have a lot of those genes that have been kind of selectively and a lot of times unintentionally, especially back in the day, genes that would allow them to interact with the local flora and fauna in a way that the current cultivars don't. So that includes mycorrhizae, that includes other organisms in the microbiome, whether it's on the leaves, on the foliage, or underground near the roots. So it's possible that by in-crossing wild populations back into these cultivars, we might be able to effectively put in resistance genes and other sorts of genetic architecture that would allow the cultivars that we grow nowadays to be more robust and be resistant to the organisms we want them to be resistant to, and not resistant, and in fact encourage the relationships of microbes that we do want to see with our plants. And these microbes can be vertically transmitted from one plant generation to the next. So sometimes we'll find that a seed actually includes many different microbes, like inside the seed, ready to be growing with the plant as it sprouts, which is a pretty amazing concept. And then they can be vertically transmitted, so from parent to offspring. That is to say, microbes can ingress inside a seed by many means, including maternally via the carbohydrate transport route from the leaves, all the way to the seed's outer coat, paternally via pollen grains, or environmentally via penetration of the nectarthodes, or the stigma style of the flowers. In addition to vertical transmission via parents, members of the plant microbiome, which has been hashtagged, the aggregate of all microbes in a unit, in this case an individual plant, are transmitted horizontally from the surrounding environment. So horizontal transplant or transmission is from peer to peer, is from plant to plant. Vertical transmission is from parent plant to baby plant to the next offspring to the seeds. Or for several wild plant species, possibly via passage through the gut of animals. Microbes have been localized inside the seed coat as endophyte organisms. Endo means inside, phyte means plant. But can also be found on the cotyledon, as well as the root hypocotyl embryo after seed germination, as is depicted in the picture, which I will have on the video when I edit it in, wherein Serratia, which is also a microbe that I keep seeing pop up in these sorts of microbiome studies, Plymuthica was used, so Serratia plymuthica was used as an inoculant for further study of this endophytism. Microbe composition of seed is diverse and can contain up to 9,000 microbial species and up to 2 billion bacterial cells in the Zea, for example. The microbial footprint of domestication in food crops like Zea, maize, and Oriza, rice, reveals a shift in some crops from slow-growing bacterial phyla, such as Bacteroidetes, on wild crop relatives, and ancient cultivars, to fast-growing phyla, such as Proteobacteria, on modern crop cultivars, necessarily changing the physiology of those plants and their collective microbiome. Seeds are sterilized externally in a number of ways to prevent pathogenic microbes from ingress. But in the last few decades, it has become clear that many microbial species have the potential to exist inside seeds. For more, check the title of this research report, which is Saving Seed Microbiomes by Berg. Right, so that's the last post in these examples. What am I getting at here? I'm just going to reiterate and summate what I was saying before. Essentially, what you can do, what I've been doing and what I'm trying to do, mostly for myself, but also for other people who want to follow along, is that when I make these hashtags for the taxonomic groups, you start to see these patterns kind of crop up, and that's what I wanted to articulate with these posts. You see, I mentioned it here with Serratia. If I click on it right now, I get a whole bunch, so I get the entire hashtag group from Instagram. This is on Instagram. 827 posts. There's a lot of Petri dishes, right? So somebody who's like, what is Serratia? What is that? Tell me more about this. There's a ton of people making art with them, which is interesting. There's a picture of a drain, some more art. University of Florida, nice. So it's not very, there's captions of these pictures certainly, but not super informative from a pictographical sense. But here's the thing. Those who know that Serratia is a facultative entomopathogen. So that means that it's an organism, a microbe, that sometimes infects insects, but not always. And it may have positive and negative effects on the host, depending on the species and the strain and all this other sort of information. So when you start, here, I have another picture about that, actually. Boom. Okay. Another post here. And what is this talking about? People typically salespeople, and this post is from June 1st, 2018. I say that people, typically salespeople, and those with a vested interest in an au natural aesthetic, like to tout the importance of biopesticides and hashtag biocontrol agents by saying something to the effect of, unlike chemical controls, pests can't become resistant to being eaten by biological controls, which is intellectually dishonest, and I still stand by that statement. Biocontrol resistance is real. Plenty of times have I now expounded on this particular topic. I find the subject incredibly interesting as both a threat we must mitigate and as an example of the adaptability of life, specifically regarding good symbioses. In the recently accepted article entitled Repressed Beauveria bassiana Infections in Delia antiqua Due to Associated Microbiota, researchers found that bacterial symbiont organisms Citrobacter frundii, Enterobacter ludwigii, Pseudomonas protogens, Serratia plymoutheca, Sphingobacterium faecium, and Stenotrophomonas maltophilia, frequently isolated from the onion fly larvae, suppressed Beauveria bassiana canidia germination and hyphal growth. Wow. So what does that mean? That means that these bacterial symbionts, so these bacteria live inside the onion fly, okay? And these bacteria were able to help the host larva. It's a pest, by the way. It was able to allow it to essentially destroy or suppress the pathogen Beauveria bassiana from infecting it. which is, by the way, an important biocontrol agent that it's like my favorite one to utilize in cultivation. It affects a wide variety of pests. It typically doesn't go after biocontrol agents that we utilize now, so it's a really nice sort of organism to have access to. But here we see that there's obviously examples where these symbiotic organisms make Beauveria bassiana less effective, otherwise tolerant or resistant. So it's very, very incorrect to say that biocontrols can't be resisted. That's just not a true statement epistemologically. In fact, reinoculation of specific individual bacteria enhanced the resistance of axenic, in other words, non-symbiont having larvae to Beauveria bassiana. So they were able to actually like vaccinate these larvae from Beauveria bassiana by taking the bacterial symbionts that did allow for resistance and just put them into the other larvae, and it worked the same way, which is pretty fascinating. So how does this happen with pests? Well, they sometimes pick up microbes just by eating the leaves. And that's my point, right? So you can essentially, like in the original post that I made, the original meta post that kind of had all the other examples, they were talking about all of these microbes that live in the leaves and live in they live on and inside the plant foliage and on and inside the roots. Well, some of those things help the pests. Some of them help the beneficials too, but a lot of them can help, in some cases, the pests. They might just pick them up simply by eating the leaves and then having those like microbes get into the body and then just live in the gut. That happens. That's how viruses are transmitted. That's how a lot of pathogens are transmitted. It's not surprising to find that this is how symbionts are picked up by certain organisms, especially if they associate on the same plant for millions of years or thousands of years or even like a few years. Like this sort of evolution can happen very quickly. And that's why I think it's important. It's important because this sort of a thing ties into the fundamentals of understanding the microbiome. It's the fundamentals of integrated pest management, having a holistic approach to combat pathogens and pests. I think it gives a great context about what people are dealing with and how these sorts of interactions and relationships, both the beneficial and the detrimental, both with the host plant and with pathogens and with pests and with the pests, the organisms, the microbes that help the pests and the ones that harm the pests, and that whole complex spider web of interactions is really important to really consider. At the very least, consider, ideally to understand. You can see in the graph, which I will post in the video, that over the span of 20 days, larval populations without these symbionts died, which is the violet line, died readily, whilst those with them survived much better, which is the green and red lines. They're practically at 100%, maybe like 90%. You can also see the microbe's level of presence in the larval gut, and if I look, I can see that in the instar1, in the level 1, level 2, and level 3 larval stages, there is pretty much the same composition. However, we see that Delphtia surohatensis, which is this like violet color, changes from the first to the second. It expands quite a bit, and then from the second to the third, it retracts back. I see that Stenotrophomonas maltophilia also kind of goes from like a little bit to a lot to greater, and Pseudomonas protogens definitely goes from having a lot in the first stage to a little bit less to the same amount, more or less. So all of the other microbes that I mentioned here are represented in the picture. The species name has been the species name has been hashtagged so that I can easily come across it if I want to. And yeah, I guess my point is that that's why I use hashtags the way that I do, and I think it's really helpful for trying to get context and trying to get information, and also just being self-referential on the internet. When somebody has like 3,000 posts or 9,000 posts or like whatever, some exorbitant amount of posts, nobody's going to go through that entire like library of information. They want to have ways to selectively go after it, and so do I. So that's what I do. And that was the reason why I wasn't answering any questions, Can of Bliss. I saw all of your comments this entire time. I'm sorry that I was ignoring you. I just want to get all of my ideas out. I knew that it would be a very long video, but I definitely saw what you were talking about. And yes, Fusariosis, to answer your question, Fusariosis is a thing that humans can get. Animals can get it too. In fact, if you really want to gross yourself out, you can look up fusarial diseases. And I think I saw on one of the hashtags there was like an infected limb from like a horse or something. It was really, really gory. And so I can see why Instagram wouldn't necessarily want to have that on their platform. It makes total sense to me. But, you know, I'm not as squeamish as other people. So, and also it doesn't necessarily mean that like people should be afraid if they're working in like a cultivation space. Am I going to get fusarium? Well, probably not unless you're immunocompromised. And in that case, you're probably not working in a cultivation facility anyways. So you won't have to worry about that so much. I am glad to hear that your ladybugs are working better than your synthetics, whichever those were. Fighting Cancer Daily says, that trope seems to be associated with acquired resistance to pesticide methods of action, likely tied to somatic mutations influenced by the mutagenic nature of pesticides in pests with a short life cycle. I have no idea what trope is being referred to, since there's no context for that, but I don't necessarily agree that pesticides are always going to be mutagenic, unless what you mean is that like pests adapt to sublethal and lethal levels of pesticides. That's definitely true. In fact, spider mite, the biocontrol one. Yeah, you're going to have to give me more context. I talked about a lot of biocontrols. But it is true. In fact, there's a process called homolygosis and hormesis. So essentially, Tetranychus urticae, the two-spot spider mite, for example, and many, many other pests are very, very good at sequestering xenobiotics. Essentially, they're able to process toxins really, really well. Some of them are innately good at this, and that's why they are generalist pests. And other ones are not innately good at this, but they ended up becoming very good at this. But those that are generalists, like Tetranychus urticae, which has like 1,200 different hosts, unsurprisingly is also quite resistant and became easily quite resistant to many pesticides thrown at it. In fact, homolygosis goes a step forward. Instead of the organism being merely tolerant, it actually makes them super fecund and like 30% more reproductive, if you can imagine that. Biocontrol is not being impacted by resistance. Yeah, I guess my memory is failing me. I'm not quite sure to what you're referring to here. Yeah, biocontrols are not impacted by resistance. Oh, I see what you mean. You mean when I said the statement that that biocontrols can't be resisted. That's not quite what I meant. What I mean is that people say that biocontrols, that pests can't resist biocontrols, but that's patently false, because of course they can. Evolution is evolution. like Beauveria bassiana can definitely be resisted. That's a great example for one thing. It has nothing to do with pesticides at all. It's totally innate to the organism, way before we were using pesticides. It's part of its genome. And in some cases, however, like I'm doing a video right now, I'm composing the script for a pest primer on the navel orangeworm moth. And it's a huge problem in pistachios, almonds, walnuts, in California specifically. And it is really, really good at not being affected by the host's various phytochemicals and defenses. And that has also given it defense against various pesticides, particularly pesticides that are based on natural products. Why is that? Well, because they're based on the same things that the pest is already really good at adapting to. So it kind of, in the long run, might not be a very effective way of getting rid of the pest. That's why inherent resistances and tolerances from a genetic standpoint might be better. But I definitely agree with what you're saying with regards to pesticides, and especially a short life cycle, encouraging resistance adaptation. Absolutely. That is a really good point, and I really appreciate somebody making that point as well, because that's definitely related to what I'm talking about here. You go on to say that you feel that, this is Fighting Cancer Daily, that you feel like that's the reason people let the idea of biocontrols as irresistible continue due to the comparison of sublethal pesticide responses. Yes, I totally agree with you. Yeah. And it's you know what it also is? I've been in the industry enough to be able to say it, almost a decade now in agriculture. I think I get to say it. Biocontrols, they had a bad rap for a long time. Like for example, copper biologicals from the Netherlands has been around for 50 years or something, or 40 years. I don't want to overstate it, but they're kind of like the grandfather of a lot of the biocontrol things, and kind of established a lot of the biocontrol industry. Not like personally or anything, not like just them. There are obviously other people around the globe. But I guess what I'm trying to say is that it took a lot of convincing, and it still does to this day, of people that, you're going to fight bugs with bugs. I don't want bugs on my plants. Are these bugs going to eat my plants? People who don't understand those sorts of things. So it's been a grueling, multiple decade, longer than I've been alive, sort of uphill battle to convince people that it's okay to do that. So a lot of people are not really, who are in the biocontrol industry, are not trying to reintroduce that ambiguity. But I think it's intellectually dishonest, kind of like what you're alluding to. It's sort of and also because it's certainly a better thing than a chemical that, especially one that is likely to target a site to an organism that will easily change. So something like sulfur, for example, really hard to become resistant to, because what kills mites and insects and fungi with regards to so the reason why sulfur is effective is because there's a physical chemical reaction with the carapace, with the exoskeleton. Really hard to be resistant to that. It burns and melts it, essentially. I'm simplifying it a little bit. Whereas a chemical that is meant to attack a certain part of the physiology, like the nervous system or something, if the pest simply evolves a different receptor that's maybe 50% of the chemical, maybe the chemistry is processed and downplayed and sequestered, right? So then suddenly you have a chemical that is 50% less effective because the target site has changed in a fundamental way. Biocontrols are way more complex than that, and you're not going to have the same sort of problem. Anyways, Instagram is telling me that I only have 20 seconds remaining. But Seymour Grass Green says, biocontrol is the only mechanism which depends upon an actual understanding of natural mechanisms of population control. That is mostly true in my opinion, but I don't have enough time to say much more. Find this on Zenthanol YouTube channel. See you later.